My name's Emily Almberg. I'm the disease ecologist with Montana Fish, Wildlife, and Parks. And I'll talk a little bit about white nose syndrome today. As soon as I have, there we go. White nose syndrome in Montana, what we've been doing to study it and what it means for our bats. But I just want to start by saying it's been really fun listening to all of these different presentations. I do most of my work above ground, and I have a lot of admiration for people that have the courage to go below ground into unknown spaces and map them and those sorts of things. And I've been fortunate enough to go on a few of these cave trips in Montana to monitor our bat population. So before I begin, I'd just like to acknowledge some of the other co-authors that have worked with us on this. This is not an exhaustive list, but Christina Smucker, who's here with Montana Fish, Wildlife, and Parks. Dan Bakken, who many of you heard from last night. I don't know if Dan's here. Dan. Justin Goody, Jennifer Ramsey, Kathi Irvine, Christian Stratton, Javi Rudolph, and Evan Grant. We'll see if this works. There we go. So white nose syndrome is a disease that's caused by the fungus Pseudogymnoascus destructans. We use the term PD for short. This is a cold-loving fungus that attacks the tissues of bats in cave environments or in hibernation sort of conditions, cool, moist environments. This fungus invades the tissues and causes tissue damage that results in the bats rousing repeatedly through the winter. They exhaust their fat reserves, they become dehydrated, and many perish. If they do survive and come out in the spring, they also have to mount an immune response and recover their body condition in order to survive that critical window. White nose syndrome was first discovered in New York in 2006. The thought is that this originated from Eurasia, somewhere in Europe or Asia, where it has existed for a long time with their bats. But upon introduction into North America, we saw pretty substantial mortality and a spread westward. In 2016, it was detected in Washington State, and that represented a big long-term jump from the leading edge at the time. And so we've had some movement along the West Coast as well. And we detected it in Montana in 2020, after many years, actually, of doing surveillance in our caves across the state. This is an interesting disease in that it requires both the host and then there's a really critical environmental piece, reservoir piece. So this schematic, you don't worry about the details, but in essence, bats come out at the end of summer. They start looking for places to hibernate. They move into caves. If the cave has had PD present in the past, that PD persists in that environment. So bats start picking it up. During that fall breeding season, they're moving around to different caves, interacting with other members of the population, and they can contract the fungal spores and the infection. When they go into hibernation, that fungus can proliferate in their tissues. It sporulates, so it creates spores. That results in the tissue damage results in the bats rousing and moving around, infecting other members of their population in the cave environment, and then shedding the fungal spores throughout the environment. If the bats manage to survive all of that and come out, they can clear the infection. And by late summer, we rarely detect PD on bats on the landscape. But the trick is that they then go back into hibernation and become reinfected by that environmental reservoir, and it starts that process again. And what's important too is that over multiple years, the reservoir of the fungus increases, and that contributes to higher initial infection loads and faster rates of mortality among some of those bats that have survived up until that point. White nose syndrome has contributed to some really massive declines in our bat populations, particularly in eastern North America. The top figure here just shows growth rates, with one being a stable population here and years since white nose on the x-axis. You can see after white nose invades, those populations tank. And for a number of our species that are cave dwellers, we've seen declines upwards of 90%. And this has contributed to the listing of the northern long-eared bat, the tricolored bat, and what will presumably be the listing of little brown bats under the Endangered Species Act. So this is a really big deal for our hibernating bats. We know in Montana that we have 7 bat species that are susceptible to white nose syndrome. Those are all the bats here highlighted in red. And we have 4 additional species that are known to at least be PD positive, though we haven't documented white nose syndrome. So that's of our 15 species in the state. So we have quite a large vulnerable population. One of the interesting things and one of the long-term questions in Montana and throughout actually much of the western US has been whether white nose is likely to play out here as it has in the east. So one of the big differences in the ecology of our bats here in Montana are that relatively few of the population that we can account for use large cave hibernacula. Our largest cave hibernacula had been Azure Cave with about 2,000 bats, but the majority of our smaller caves that are known to house bats have under 50 individuals, and that's orders of magnitude smaller than what we see in the eastern US. And so our bats are using alternative winter roosting habitat, and we think some of that is probably talus slopes, some of it might be rock crevices. Honestly, we don't exactly know. They're really hard to study and follow when they go into hibernation. So that's led to a program designed to answer 2 key questions. The first is, how will PD and white nose syndrome affect our bat populations here across the state? And to answer that question, we've been doing systematic surveillance to try and understand the timing and distribution of the fungus, PD, and the disease white nose syndrome across Montana. And then we're also using acoustic monitoring methods to understand how bats are responding in terms of overall populations through occupancy and activity. The second big question is, what can we do about it? How can we maximize our bat populations here in the face of white nose syndrome? And this has really been an effort it's currently happening and underway with the U.S. Geological Survey, and it involves a structured decision-making effort. So I'll talk a little more about both of these efforts underway. So the first is our PD surveillance. We've been doing white nose and PD surveillance actually for quite a while. And I think for many years, the Heritage Program and the state were looking for symptoms in bats and caves. We started a much more systematic effort in 2019, where we collaborated with the National Wildlife Health Center, who's been asking various states across the country to survey areas along what they think is the leading edge of the fungus in a given year. And then we've taken a more systematic approach where we have a 36-cell grid across the state, and our goal is to actually survey a site within each of those grid cells to try and track the fungus as it moves. This involves targeted sampling in March through early June, and then opportunistic sampling of dead bats in winter or early spring. So this time of year is when we start to see sick bats on the landscape as they're coming out of hibernation. We use a variety of methods. We do survey caves, and a number of you here in the audience have helped with that effort, so thank you. And we sample hibernating bats directly or some of the cave substrates and cave walls for the fungus. We also do summer live trapping with mist nets, or there've been attempts to sample these bat boxes by creating a big chute and allowing the bats to fall into a big pillowcase where then we can swab individual bats. We also collect guano at known roost sites in this early time of the year to look for that fungus. So that's the surveillance piece, to try and track where the fungus and the disease is in our bat population, but we're pairing it then again with this acoustic monitoring effort that allows us to track what bat populations may be doing in response to the disease. And this involves setting up these stationary detectors, acoustic detectors on the landscape, where they passively record bat calls at night. And then we run those calls through a software program and in some cases hand vet them. And we use the signature, that spectrogram of their calls to identify species or identify it to groups of species. We try and survey 100 cells across the state, and in each one of those cells, we're putting up detectors in 4 different locations and leaving them out for 4 nights. So this is a really massive effort, and it spans the entire state. It's also in line with what other states are doing as part of the North American Bat Monitoring Program. And so those are the general methods. So now to just cover some of the results from both of those efforts and how we're trying to tie that together. This is a map of our surveillance efforts. Sorry, I don't have a legend here. But each one of these little dots are places where we've surveyed for the fungus and the disease on the landscape. And that was just last year, so there's quite an effort. All the green sites, which tend to cluster on the western side of the state, are negative for the fungus. All of these that have red and tend to cluster on the right side have been positive. The shading here of these counties are counties that we know PD has been detected. So the whole county is presumed PD positive. And some of these hatch marks highlight counties where we've detected the actual disease. So there's a difference between the disease, detecting the fungus, and the disease. Usually the fungus is detectable 1 to 2 years before we start seeing symptoms on bats, and we think that's related to having a large enough environmental reservoir and a high enough dose of infection to then cause that disease that we see. So it has made its way across roughly half of the state to date. And unfortunately, Azure, which had been our largest, as I mentioned, our largest known hibernacula of bats here in the state of about 2,000 little browns, crashed in 2022 down to about 30 or 40 animals. And we went back again this year, and similarly, there are about 30 or 40 animals. So we've seen about a 98% decline in that cave, which has been very consistent with what they've seen for little brown bats elsewhere in the eastern part of the US. So in that sense, in our caves, it doesn't look like it's playing out any differently. But again, most of our bats can't be accounted for in our caves, so we're still wondering how this is gonna look for the rest of our bats. For acoustic monitoring, we surveyed 88 cells last year out of the targeted 100. We're doing this again this coming year, collecting data. It's a huge effort across a number of different agencies and partners. And we're now at the stage of working with Christian Stratton and Kathi Irvine at the U.S. Geological Survey to integrate the information that we're getting from the surveillance, so where the disease and where the fungus exists, and all that acoustic data to see, are we actually seeing declines in activity and distribution of our bats? And this work's ongoing. The spoiler alert is that in the eastern part of the state, it does look like we're seeing noticeable declines, but we haven't yet characterized the extent of those declines. So the question is, what are we going to do about it? There have been a lot of great people working on solutions, and if you think about the problem, there's sort of 3 broad approaches. The first is to try and target PD or white nose itself, try and attack the pathogen. And that involves things like treatments through chemical, probiotic, and mechanical efforts. So things like using various chemicals to disinfect the walls of cave hibernacula, using UV light, or modifying temperature of some of these hibernacula to make it less favorable for fungal growth. There's a vaccine that's being piloted and seems to show some efficacy. And of course, there's prevention, trying to reduce human-assisted dispersal of the fungus on the landscape. Bats are doing it themselves, but we're trying to minimize that rate of spread. So those are all methods targeting white nose. They do require most of them require being able to get our hands on bats, either capturing and administering a vaccine or getting into a hibernacula environment that houses a significant number of bats. And in Montana, our options are sort of limited on that front. The other 2 ways in which you can think about trying to mitigate the impacts of disease are trying to generally bolster the bat population. So what else can we do to improve bat performance in general on the landscape? And those are general management strategies that include habitat restoration, protection, enhancement, enhancing riparian forests, expanding forest or improving forest diversity, expanding access to water sources in really arid environments. And then the other approach is to see if we can actually bump up reproductive output, right? So if we're seeing a lot of mortality to disease, if we can sort of stabilize those populations that are alive, but then also improve their reproductive output, maybe that generally helps the population trajectory. And so we're looking and exploring ideas for supporting and protecting maternal colonies through either enhanced foraging opportunities, or there's even people looking at like microclimate controlled bat boxes that are actually heated, so then when bats bats come out in the spring and they're trying to recover from the infection, it gives them a boost in recovering and reproducing effectively. So this is sort of a sampler of all the different potential ways in which we might address the disease and manage for the disease in Montana and elsewhere. And right now we're in this process of a structured decision-making exercise where we're trying to evaluate what are our best options, what are all the trade-offs, how do we, which of these is likely to have the biggest impact on our populations. And we're doing that through a structured decision-making exercise, which essentially is a standardized way of asking and posing a question, identifying those objectives, evaluating all the different possibilities, making predictions about which are likely to pay off, and then looking at trade-offs. So we're in this process. We hope to have an answer about how the state of Montana will work with the various land management agencies by the end of the year to try and bolster our bat populations and maximize their distribution and abundance. And I guess I'd just like to close by saying there's still a couple of things that we know we have we can influence now, and prevention is key. I think in the past, maybe Lori Hinniska-Brown and others have spoken, maybe Dan, to the group about disinfection protocols. And this is something that we're still advocating, and that is we're working with this decontamination protocol that's put out by the National White Nose Syndrome team. And they take sort of they provide two general recommendations. The first is try and minimize risk by keeping gear that you've used in PD positive areas in those PD positive areas. Don't move gear from known infected caves where bats are, spending time to caves that are negative or of unknown status where we do know we have hibernating bats, because we know that those fungal spores can be transmitted, can move around, and can potentially cause onward transmission. So trying to minimize the movement of infected gear. And this is the state map to date. Many of these places in the east are known to be positive. We think the majority of these western sites are negative, but we don't know the timing of when the disease will make the jump. So that aside, there's still recommendations for decontaminating gear, and those involve a number of chemical disinfectants or hot water baths of gear and clothing, which effectively kill the fungal spores and help reduce the risk of transmission. Then lastly, because I'm with the Wildlife Health Lab and we're talking about bats and caves, one of the other things that we think about in the larger bat conservation community has thought about is trying to minimize the risk of spillover of COVID to bats. And the sort of general strategy or general request of both recreationalists and researchers, and on the research side we're doing this as well, is to minimize the time that we're spending in close proximity to bats. We're assessing whether anyone is sick or has COVID symptoms or has had recent exposure to someone with COVID before deciding to go into a cave with bats, and if you have had any of those close exposures, to avoid doing so. And then if you do have to be around bats while spending time in caves, or if you're in an enclosed space where bats are using the area, using a mask is helpful as well to minimize that risk. And that's not only to protect the bats, but it's also the long-term game is we don't want more animal reservoirs of COVID, right? We don't want additional opportunities for the virus to evolve and spill back over to humans. So. I'll just close by saying this is a massive effort across lots of different agencies, and in addition to the agencies, both federal, tribal, and state, we've worked quite a bit with private landowners and many of you. So thank you. And it looks like I have a little time, so if anyone has questions, I'm happy to answer them. Yeah. The question is, does PD negatively affect any other animals? Not that we're aware of right now. This seems to be restricted to bats. So the fungus likes, it's most effective at growing under cold temperatures, cold, moist temperatures. And so it does invade, mammalian tissues, but the tissues have to be cool, and it has in order for it to thrive. And in, bats, when they're in torpor or in hibernation, their immune system also slows, and so it creates the perfect opportunity for that fungus to thrive on their skin and proliferate. But yeah, no, look, it's not and in fact, when bats wake up and they're warm and they spend time outside moving around, the fungus is not an issue for them at that point. It's just during hibernation. Other questions? Yeah. Yeah, that's a great question. The question is, how did it get to North America? Was it via bats? Oh, but Azure. Sorry, Azure. The assumption is that it's bats. So the wave of infection had been, hit the eastern part of the state. We first detected the fungus in, I think, 2020. And then saw symptoms 2021, and then the full-blown mortality event by 2022. So yeah, bats are moving it around. It's, one of the interesting things is that it's taken longer to jump the continental divide and into the western, part of the state than anyone expected. When we first got the predictions from the National Wildlife Health Center, they were suggesting that, it was going to be jumping a number of years ago now, and it's taken longer. And so we don't know why that is. It could be the population structure of the bats and where they're spending time and how they're moving and interacting with neighboring populations. It could be something about the roosting ecology we don't know yet. So, yeah.
The other side of the divide seems pretty clean right now. Do you know where, kind of the front coming off that Washington infection is at?
The question is, where is the front from? From Washington. So this map, actually, if you have questions, this map is publicly available at whitenosesyndrome.org, and they keep it updated. So as new detections appear, they'll add counties to the map. There've been detections here in the south. Those are probably from the west, and I think the assumption is that maybe some of these are related to the Washington event, but we don't know for sure. So it seems like the state is still being covered pretty much by that eastern wave, moving westward. Yeah, Ellen.
So obviously, as white nose becomes pretty prevalent throughout the state, and it's a weird patchwork of some places are positive, some places negative. Do you recommend that cavers go by county level or specific cave level as far as what's the population that's in it? That is kind of difficult to navigate.
Yeah. The question was, as white PD and white nose becomes more prevalent across the state, how do cavers make the decision about what to do given whether the cave is of a known status, positive or negative, or if it just falls within the a county that's known to be positive. There's always some uncertainty with our ability to classify the status of a cave. Part of that is whether we get back to it regularly. And so I think generally the recommendation is still use the general county level information, just recognizing that it's imperfect. And so when in doubt, if you can have gear that's somewhat dedicated to a particular area or do a really thorough job of disinfection, that's probably my best recommendation. I don't know. Dan or Christina, do you have additional thoughts? Yeah. Yeah, Yeah. Yeah. To the community, yeah. Yeah, certainly. We do publish an annual report, but it's usually at a lag, so there might be some ways to think about, posting some of those results on our website more in real time. But yeah.
So we're doing our sampling right now, and probably by the end of the summer or fall, we'll have the results from Whitaker and Lick Creek and some of those that are still, east of the divide, kind of starting to get surrounded by white coats. But I mean, for this season, I would almost say like east of the divide, west of the divide might be the safest thing if
Yeah.
you're thinking about caving. Like Jan said, we get a caving gear is expensive, but thinking about keeping your gear separate if you're able to, or just getting crazy if you're going to see that.
Yeah, and it's good to think about outside of our state borders too. I don't know how many of you spend time caving in the other neighboring states, but again, this is probably the best resource, this map on whitenosesyndrome.org, to look at where it's been detected to date, so that you have that information for some of our neighboring states. Yeah.
How does the Montana surveillance program compare with other states and some of the main provinces? Are they as active, more active?
So the question is, how is Montana's surveillance program compared to other states? There is a fair bit of surveillance that's being done in other states. Many of them have been partnering with the National Wildlife Health Center, which again has been tracking that wave. So there's been good national participation in that effort. I think I'd say maybe we're doing a little more than most. Yeah, we've been doing a little more in terms of filling in the gaps behind that wave that haven't yet been confirmed, and trying to keep it going systematically and having a really broad survey effort. One of the challenges, like it's one thing to detect the fungus and track the fungus. It's another to track the actual disease, the appearance of lesions in bats that are being affected by it, and that's more time intensive. It requires netting in that early spring to look for lesions on bats that have emerged, and or going into caves with known bat populations. So that part has been a little more challenging. I'd say that's patchier than our ability to detect the fungus as it's moving. But again, we're just trying to do the best we can with the survey effort, and then pairing that with some of the acoustic information, which at least gets us a handle on trends and what our bat populations are doing and who's being impacted. So, yeah.
Is it basically just you get one big die-off and then behaviors change or immunity develops, or do you keep getting die-offs as the population gets back up?
The question is, do you get one big die-off and then immunity occurs and things stabilize, or do you see recurring outbreaks or epidemics? The pattern to date has been, as I said, usually when the fungus arrives, there's about a 1 to 2 year lag before there's enough of a buildup of the fungus in that environmental reservoir, such that bats coming in are infected with high loads, the fungus proliferates really extensively, and then they succumb. It varies by species, so some species are way more susceptible than others. And generally, it's that first big crash. There are some that continue to persist. Many have blinked out entirely. Small, some of the small populations, which initially we had hoped, like maybe there's a smaller density, so transmission would be less pronounced, it'd take longer to get there, they tend to blink out. They tend to just disappear, unfortunately. And there's been a lot of work studying trying to understand why certain individuals are persisting. Some of it is intense selection on, the genetics of that individual that probably modulates the immune response. So that's an ongoing area of inquiry. Yeah.
Can you talk a little bit about those few parts of some of those sites that are stabilized and maybe start to come back?
Yeah, I don't know that I have. So this figure on the bottom, which I just glossed over, are a bunch of different species of bats. A number of Myotis species, big brown bats, tricolored bats, and this is the percent change after white nose syndrome's arrival. So 0 is where they're, if they're stable with no change, and then these are the big declines. And you can see there's a lot of variation across, these are states and sites within a state, so you can see a really big range. And yeah, as Christina indicated, there are a few sites in the east where they're seeing it stabilize. Again, for many of them, they're at like 2 to 5%, maybe 10% of their historic numbers, but at least they're not continuing to decline at this point, and the hope is that long-term those will continue to build. As I said, there's active research on using various intervention strategies, particularly in the east where they can get into these big hibernacula and try disinfection, for example, or UV light. But I think everyone recognizes those are tools, but they're difficult because they're labor intensive. They're sort of, in many cases, a forever sort of treatment, or maybe until the population starts to do a little bit better. So it's tricky. There's no silver bullet. It's a tricky one.
How long can the spores survive in a sealed cave?
The question is, how long can the spores survive in a sealed cave? That's a great question. I don't know if I know the answer offhand. It's certainly over the year they can survive. There is a decay. There can be a little bit of a decay sometimes, but generally we're seeing persistence for multiple years, and that's probably something that's being studied. So for some of these caves that have seen the complete loss of their bat populations, they can track that in persistent reservoir and see how long it survives. But it's on the order of 1 to several years, probably at least. Maybe even longer. Yeah, maybe even longer. And the thought is that sometimes there's cave flora that may help eventually contribute to the degradation of those spores, but yeah, I don't know. There's some really great research on European cave systems too, and Asian cave systems, and PD, the dynamics there. And they do see a more pronounced decline in the environmental reservoir over the summer months. And they think that's pretty important for bats being able to survive without getting really bad lesions. But presumably, way back in history, whenever PD emerged in Asian and European caves, there was probably some large die-off event. And some have speculated that, the abundance of bats that you see in European caves and Asian caves is now more similar to sort of the abundances of bats we see in the eastern US. I mean, it's like, before that, we had orders of magnitude more bats in some of our hibernacula than they would see abroad. And so one of the hypotheses is that historically PD completely reshaped the populations of bats on those continents. So. The fungus does best in cool caves with relatively high moisture. But again, there's a fairly wide gradient where PD has been found, and that is, I guess, the one comment I'll make on that is that there does seem to be a southern extent. In that the fungus, as you move farther south, caves become warmer, and bats spend less time in hibernation. And those two things combined seem to create this barrier for white nose, causing problems for many of those southern species of bats. And the hope is that that those tropics will be an effective barrier. The concern, of course, is the potential for human-mediated, or the movement of bats from the northern hemisphere to the southern hemisphere, where there are potentially caves in the farther southern hemisphere that would be suitable for PD. But right now it does not exist in the southern hemisphere. Yeah. Have we noticed any ecological changes related to declining bat populations? This is an area of active research. People are interested. There's been, I think there's one study that's looked at tying the purchase of pesticides to some of the concurrent declines in bat populations. But I think this is sort of an area of active and ongoing research. But of course, the concern is that, these bats are huge, huge consumers of pests, agricultural pests, and that with such a huge shift in their population sizes, we could see big releases on a number of our insect pest populations that would impact agriculture. Yeah. All right. Oh, do I? Okay. Yeah. Yeah, so the question is, have there been studies on the efficacy of various chemicals and various treatments for killing the spores? So yes, everything in that decontamination protocol has been vetted in laboratory and sort of field conditions. So there's things like bleach solutions, various disinfectant chemicals that are effective. In terms of what's been effective in caves themselves, that's an area of active research. But yeah, we just listened to a presentation on using sort of like a bleach fog in abandoned mines that are used by bats in the summer when the bats are gone, but going in and essentially disinfecting those cave environments. And they have showed they can knock back PD, and it can have positive impacts for survival for the remaining bats. So there are some tools like that. Again, labor intensive. And in some cases in the east, those declines have already happened. So, so much so that, I think states are weighing the relative merits of going in and doing repeated treatments at this point versus just trying to support remaining populations. Thank you. Thanks for all the questions.